Yes. Now, if you recollect, last lecture we were talking about menstrual cycle, in which we have learned the first menstrual cycle is called as menarche, while the last menstrual cycle is called as menopause. Now, I just tell you how the first menstrual cycle starts. When a girl child reaches at the age of around 12 to 14 years. In that girl child, hypothalamus start producing gonadotrophin. Releasing factor. This gonadotrophin releasing factor act on pituitary. Pituitary start producing FSH and LH. That FSH and LH causes development of follicles. These developing follicles produces estrogen. When this follicle mature, they undergo a process called ovulation. From ovary, ovum is released, which goes into the fallopian tube, and a special endocrine structure is formed, which is temporary, called as corpus luteum. That corpus luteum produces progesterone. Progesterone causes progressive thickening of this endometrium. Now, since girl is at the age of 12 to 14, definitely there will be no chance for pregnancy. Now, if she is not becoming pregnant, then definitely after 7 to 8 days corpus luteum activity is reduced, the progesterone levels start decreasing. By the end of 28th day, its level become lowest. So this endometrium start falling off. When this endometrium layer falls off, definitely blood start coming out through vagina. That is the first menstrual cycle. Now it will become a periodic event. Every month the same thing will be repeating. Clear? Now please understand, in this video we are going to talk about an event which can disturb this menstrual cycle. Means an event which we call it as pregnancy. Now for pregnancy, the first step is fertilization. So today we are going to talk about very important part of pregnancy, that is fertilization. Now the first step of fertilization is insemination. See, semen is released into the vagina during copulation. Now, if semen is released into vagina during copulation, we call it as natural insemination. There is also a technique which is called as artificial insemination. We'll talk about it afterwards. Now, when insemination takes place, roughly around 3 to 4 ml of semen is released, which has around 300 million sperm. Now, once the semen is released, sperm start migrating. Check. Okay. When sperm is moving, they undergo a process called as capacitation. Capacitation is chemical maturation of sperm. Most of the maturation of the sperm complete in epididymis. But remember, capacitation is a process which is must for a sperm to become capable of fertilizing the ovum. Now, what happens during the capacitation? Now, this I have drawn a rough diagram of sperm. If you recollect, this is a plasma membrane, this blue one membrane. The red part which you are seeing is nothing but the acrosome. This black part is a nucleus which is haploid. This dot anterior one is called as proximal centriole. The lower one is called distal centriole, and then axial filament. These are mitochondria. Yes. Now what happened in capacitation? Because of certain chemicals from fallopian tube and you can say certain hormonal factors causes the membrane, the surface membrane of the head of the sperm become weak, so that this acrosome will be able to fuse with the membrane and release its chemical when the sperm wants to enter the ovum. So hope I am clear with you the part called as capacitation. So remember, first step of fertilization was insemination, the semen was released. Second process was capacitation, which is nothing but chemical maturation of sperm. Here the head region of the sperm gets partially, you can say, protein get eroded, so that acrosome will be able to fuse with the head and anterior part of the head and release its chemical. Now sperms are moving. During this journey, they come across physical barrier. They also come across chemical barrier. Chemical barrier means female vagina is highly acidic, so most of the sperm die. They also come across macrophages, WBCs of the female body, which considers sperm as a foreign body, and they destroy them. So out of this 300, sometime it could be 400, sometime it could be 200 million sperm also. Out of them, only 200 to 300 sperms are able to reach the ovum. That means, what I mean to say, that almost 1 in a million sperm is able to reach. So that means, remember, all the person who had born, that means at least 1 in the million case you had won. Okay. Now the sperm is reaching the ovum. Now please understand here, it's not that sperm will go only on right side, it can go also on left side. Wherever ovum is there, that side it will fertilize with the ovum. If ovum is not there, then these sperms are destroyed by macrophages and they die. So now let us consider these sperms have reached the ovum. Now when they reach the ovum, please understand what happened here. In this here, when the sperm is approaching the ovum, a kind of chemotactic movement takes place. Here a kind of reaction takes place, which is, we call it as fertilizin-antifertilizin reaction. Now what happened? Certain sites on zona pellucida, which already we have learned, blue membrane mass, it's a non-cellular protein layer. It has certain receptors. We call those receptors as ZP3, zona pellucida 3 receptors. There they have certain ingredient which we call it as fertilizin, which causes chemical attraction to the sperm, because sperm has a chemical called antifertilizin. This fertilizin and antifertilizin bring about a confirmatory change, which is responsible for integrity of the species. When I say integrity of the species, means what? If this is a human ovum, the sperm of human only can fertilize it. No other sperm will be allowed to enter, because that reaction, which reaction? Fertilizin antifertilizin reaction will not be possible. That's why we say this step checks the compatibility of species. Now let us consider sperm had reached the surface of the ovum. The moment it reaches of the surface of the ovum, let us consider this had reached the surface of the ovum. Immediately a step to start, which we call it as acrosomal reaction. Acrosomal reaction means what? I told you now, the sperm head undergoes capacitation. So this part, that means anterior of the head of the sperm, fuses with the acrosome. So see here, it had open, so start producing chemicals. Chemicals start coming out, see. Now what are these chemicals? First chemical is hyaluronidase, which dissolves hyaluronic acid. Now remember, hyaluronic acid is a polysaccharide which holds the corona radiata cell. Now because lot of sperms are releasing hyaluronidase, the corona radiata cell loosen up and they are start separating. Now because of this, sperms are able to reach the zona pellucida. Once it reaches the zona pellucida, sperm produces acrosine. Which is also called as zona lysine. This zona lysine bring about digestion of zona pellucida, and now sperm is able to move into perivitelline space zone, or I can say it is able to touch vitelline membrane. Now see, perivitelline space is showing you first polar body. That means this structure is still secondary oocyte. The moment sperm touches the zona pellucida ka inner part, that means it reaches the vitelline membrane. Now this cell initiate the meiosis 2 process. Meiosis 2 process will trigger here. Is it clear? That means cell is undergoing now, means it is about to complete its meiosis 2. So what happened here? A small receptor cone, fertilization cone, is formed in the ooplasm. Head of the sperm now almost in contact with the membrane, pushes the male nucleus inside, followed by proximal centriole. Now the moment it happens, simultaneously I can say the moment nucleus had enter, here meiosis 2 completes and we get a second polar body. If you see, only the anterior part of the head had entered into the egg. Now I am using the term egg. Till now it was secondary oocyte. But can you see now 2 polar body? Since 2 polar body is formed, we can call it as egg. Nucleus had entered, proximal centriole had entered, remaining structure remains out. Now remember, this were all we were learning under the heading acrosomal reaction. Now here very important step take place, which we call it as cortical reaction. Now what is this cortical reaction? Remember, this cytoplasm shows large number of cortical granules. The moment one sperm enters, they plug the opening, cortical granule plug the opening. Second, the membrane, the egg membrane, that is vitelline membrane, undergoes depolarization. This depolarization event prevent any other sperm to enter. So it's kind of a block is created. Not only that, by the time these cortical granules releases certain chemical which destroys all the receptor on the zona pellucida, that means now even zona pellucida becomes impermeable for the sperm, because sperms were able to enter only through receptor ZP3. Now they are not there. Why? Because that chemical had destroyed. So now zona pellucida becomes impermeable for any sperm. Student asked me doubt, suppose sir, some sperm might have entered into perivitelline space, but this had entered, now it is just outside, then also it will not be able to enter. Some students say, sir, if two sperms directly enter at the same time, not possible, because remember, if two sperms enter, then two sperms nucleus will have 23 plus 23, 46 plus female nucleus 23, 69. Human, not possible. So in such cases the egg will die, but practically it is not possible. Only hypothetically I can say. Only one sperm enters. So we say that the cortical reaction prevents polyspermy. Hope I am clear with this. Now the nucleus which had entered travels straight. During this path we referred it as male pronucleus. Female egg nucleus is called as female pronucleus. The nucleus attract and they fuse. So nucleus is traveling here. Now they are attracted. See, it is bending. And finally both fuses. Now remember, when it is traveling, head region, nucleus make a turn in such a way that it will be easily to fuse with the female pronucleus. And once the fusion is completed, so we have a large nucleus with 46 chromosomes, 23 from father, 23 from mother. So now this we call it as zygote. The entire process of fertilization takes place roughly around 8 to 10 hours after sexual intercourse. Remember, major time period goes for capacitation, which takes around 4 to 6 hours. Without capacitation, sperm will not have capacity to fertilize. So I just brush up again. What are the steps? First step was insemination. Second step was capacitation. Third step was fertilizin-antifertilizin reaction. Then there was acrosomal reaction, due to which sperm was able to penetrate. Next, after that there was cortical reaction, which major role was to prevent polyspermy. And finally there was SYNGAMY, where male and female pronucleus fuses to form diploid zygote. Clear? Thank you. Now remember, this part is very important, especially for NEET point of view. I have seen, at least if we consider last 4 or 5 years, at least 3 times MCQ was there. One of the most important question comes is that when second polar body is formed. To this I can easily say that it is just before fertilization, after the entry of this sperm. Yes or no? Yes. Next, the chemical which is produced, which helps in separating out corona radiata, was hyaluronidase. Zona pellucida was dissolved by zona lysine or acrosine, which is produced by nothing but acrosome, which is nothing but a modified Golgi complex. Now remember, compatibility of this species is done by fertilizin and antifertilizin reaction. Now remember, the sites I told you, specialized receptor on zona pellucida, are responsible for it, for facilitating the entry of sperm into the ovum. Remember, when the process starts, it starts in ampulla. Is it clear? An entire process is completed there. So if I ask you question for site of fertilization, then you can easily say it is nothing but the ampulla, or you can say fallopian tube. Remember, major changes which bring about in fertilization is, first, haploid structure changes into diploid zygote, and also proximal centriole makes an entry. Centriole, which was not there, had entered into the egg. So zygote has a centriole coming from sperm. Many sperms try, only one is able to fertilize it. So recondition is called as monospermy. We are able to prevent polyspermy because of this fertilizin membrane, and also because of the depolarization of egg membrane. With the fertilization, first step of pregnancy had initiated. Now remember, the ovum which is released is generally active for 24 hours, max to max. Extended period could be 48, but generally it is active for 24 hours. So generally fertilization take place on 14th day of menstrual cycle. Clear? Now, once the fertilization is done, next step, which we call it as cleavage, will be initiated. Fine? Okay, we'll stop here.